Billy, and our group will be talking about Clemson University squirrels today. So the species involved in the study is the eastern gray squirrel, Squirrus carolinus, and the habitat is mixed hardwood forest and urban/suburban areas, such as parks and college campuses, and also suburbs. And the eastern gray squirrel has grayish brown fur, a white belly, and pale gray to brownish bushy tail. And the key behaviors for eastern gray squirrels are foraging, so collecting nuts, seeds, and fruits, vigilance behavior, which is increased alertness to potential threats, especially around human activity, and social interactions. And squirrels communicate with things such as tail flicking, dominance displays, and interactions with other squirrels or other species, such as dogs or people or any other animals. So the objectives of the study are objective one is to calculate the frequency of vigilance and social interaction behaviors in squirrels during periods of high and low human activity. Our second objective is measure the duration of foraging behavior in squirrels during periods of high and low human presence. And objective three is compare the frequency of vigilance and social interactions, as well as the duration of foraging between areas of high human activity and low human activity. Our test hypothesis for the study was squirrels on campus will present different behaviors such as foraging, vigilance, and social interactions in response to the presence of humans, and an increase in vigilance and a decrease in foraging when there are increased activities of humans nearby. And I wanted to talk about some relevant studies that we found. So the first study was differing vigilance among gray squirrels along an urban-rural gradient on Long Island. And the key findings here is that the study examines the vigilance behavior of gray squirrels across different landscapes, focusing on how squirrels in urban environments may be more vigilant due to higher human presence and potential threats. And the study shows that squirrels in urban areas tend to be more alert, possibly due to the greater frequency of human interactions and predator risk. And the relevance of this study is that it directly addresses how urbanization influences vigilance in gray squirrels and sets the stage for understanding how human activities can impact squirrel behavior. For the second study that we looked at, it was for seasonal comparisons of daily activity budgets of gray squirrels in urban areas. And the key findings in the study that the study compares the daily activity patterns of gray squirrels in urban settings during different seasons. The study shows that urban squirrels may change their activity patterns, particularly in terms of foraging and social interactions, to adjust to human activity and the challenges of living in an urbanized area. And the relevance of this study is that it's important for understanding how gray squirrels change their activity budgets, such as time spent foraging or avoiding predators, in response to environmental changes associated with urbanization. And for the third study that we looked at, it was for behavioral responses of eastern gray squirrels in suburban habitats differing in human activity levels. This study highlights how gray squirrels in suburban areas show behavioral differences based on human activity levels. In areas with more human presence, squirrels may alter their foraging patterns or increase their vigilance to avoid potential threats. And the relevance of this study is it helps broaden our understanding that gray squirrels adjust their behavior not only in urban areas, but also in suburban areas, suggesting a range of behavioral responses based on human activity levels.
The methods. This behavioral study was conducted at Clemson University in multiple locations on campus where squirrels are frequently observed. To examine how human activity influences squirrel behaviors, we use direct observational methods. These are non-invasive and great for wild animal behavior studies in urban environments. Study sites and timing. High human activity areas were near Pool, Martin Hall, the Botanical Gardens, and walkways. Low human activity areas were near Fort Mill, botanical gardens, and less traffic green spaces. High human activity periods were during class time changes, university tours, and lunchtime. Low human activity periods were early mornings, late afternoons, and evenings where campus had less traffic. And here are some images of the squirrels. And here are the locations we observed them. Sampling and observing protocols. We used focal animal sampling where we selected one visible squirrel at a time and observed it for 10-minute intervals. During these sessions, we recorded many behaviors. Vigilance, which was looking around and standing alert. Social interactions, which was chasing, grooming, vocalizing with other squirrels, and foraging, looking for, handling, and consuming food. Behavioral data collected, frequency of vigilance and social interaction, durations of the foraging behaviors. And then we had sampling goals. So our goals were to have at least 20 to 30 observations per human activity category. Observations were done over multiple days to reduce bias. And lastly, data analysis. Calculate the mean durations, compared frequencies, and we used ANOVA to compare the differences between high and low human activity. Ethogram. And lastly, here are some videos of foraging, vigilance, and social interactions.
For our results, this chart presents the total number of squirrels seen, the vigilance frequency in low, moderate, and high human activity areas, and the social frequency also in those activity areas. This graph is a visual representation of vigilant behaviors related to human activity. This graph is a relation of social behavior related to human activity. As we can see, more social behavior was conducted in higher activity areas. This chart shows human activity level compared to total foraging time, the average foraging time, and the percent foraging time uh, that was collected. And this graph relates the total foraging time in the different areas to the mean time of the different areas. Finally, an ANOVA was conducted to compare the correlation between the behavior seen and the human activity level. To be significant, a p-value must be less than or equal to the alpha, which in this case was 0.05. So vigilance behavior had a p-value of 5.87 to the times 10 to the negative eighth, meaning it was very significant because it was much lower than the p-value, where social behavior and foraging time were higher than the p-value and therefore not significant.
So, for conclusions, the interpretation and hypothesis. So, our hypothesis was supported. As you could see with the results, vigilance increased significantly under moderate and high human activity compared to low human activity, and remained high during high activity, suggesting sustained alertness. And then foraging times decreased from 88.33% on the low to 36.36% on moderate and 53.68% on high. Social interactions increased, which was a little bit different than, I guess, what we expected, under high activity, suggesting maybe a complex social or stress-driven response. Um, so comparison with previous research. So with our first research, our results supported their conclusion that urban squirrels exhibit increased vigilance in response to human presence. As far as the second paper by Parker, our findings indicated comparable changes in foraging behavior influenced by environmental factors and human interactions. And the last paper validated that squirrels modify both their vigilance and foraging strategies in relation to human activity levels, aligning with our observations as well. Um, so enhancement for future research, something we can maybe have done a little bit different. So utilize a larger sample size and diversifying the locations of studies, analyzing individual squirrel identification to gain insight into reoccurring behaviors. That's a little bit harder because we don't really have like technology to do that, or it would be hard to kind of like track the squirrels and like tag them. But if you could, it would be very interesting. The third one is to ensure constant recording durations for squirrels during each data collection session. And the last one is maintain a uniform number of squirrels observed in each data collection effort. And this is our literature cited.